Hi, I'm Kathleen Treseder from University of California, Irvine, and I'm excited to tell you about our research on how evolution and trait trade-offs in fungi could mediate carbon cycling under climate change. I want to start off by acknowledging the lead PI of this project, Steve Allison, and all my co-PIs who are listed here. It's been a really great collaboration. I also want to acknowledge that as I'm recording this, I'm sitting on the land, the traditional land of the Hashkavan and Kitch nations. And they're still here, they're still very much active and working on the environment. And I want to make sure that we recognize that. And the work that I'm going to tell you about is part of a larger project. This is the project that DOE is funding. And the overarching question for this project, the larger project, is how does evolutionary adaptation influence microbiome response and functioning under drought conditions? Um, we now have a lot of data on how communities can shift in response to climate change. We have data on how the physiology of microbes may change, but we have less information about how microbes might evolve in response to climate change, especially in the field. Now, this is something that we shouldn't overlook because microbes have fast generation times. They may be able to respond relatively quickly in an evolutionary sense to climate change. And the system that we chose to address this question in is coastal sage scrub and grassland in Southern California. These ecosystems are endangered. There's not very many of them left. And in addition, they're already fairly arid. So if we have increased drought here, which is predicted by climate change, we might be pushing these microbes up against their climate limits. And in particular, we're testing this trade-off framework that was proposed by Ashish Malik when he was a postdoc in Steve Allison's lab. With this trade-off, we're thinking about three major functions that fungi can perform. There's stress tolerance, the ability to produce biomass, and the ability to acquire resources from the soil, specifically by releasing extracellular enzymes that break down organic material. Okay, so the idea here is that if a fungus invests in one of these functions, like say drought stress tolerance, then that means that there are fewer resources available to invest in these other functions like biomass yield or resource acquisition. And that's just because resources are finite. So these trade-offs we expect might set up fungal communities like right now in an area in which the taxa vary in the extent to which they perform each of these different functions. You could have one taxon being particularly good at one of these functions but poorer at the rest, or you might have a taxon, say, be mediocre at all of them. Okay, but you're not going to have one that's good at everything. So now the question is, does this happen? And we can use a relatively new technique to answer that question in the field. And we can do this by using what we call microbial cages. And this was developed by Jen Martini. So these cages are basically envelopes that are made of a nylon membrane with pores that are very, very small, about 0.42 nanometers diameter. Those are too small for fungal hyphae to grow through and for most bacteria to pass through. So this means that if we put a microbe inside these cages, that they are stuck there. In addition, other microbes can't move into the cages. Right, now, these pores are actually large enough for water and nutrients to pass through. So this means if you put a microbe on sterile litter inside these cages and place the cage in the environment, it will experience the moisture and nutrient conditions that are typical for that area, but we can isolate the specific response of that fungus in the field. So a former postdoc in my lab, Charlotte Alster, used these microbial cages to test for trade-offs among traits in fungi growing in the field. Okay, and she did find evidence for trade-offs between drought, stress tolerance, and resource acquisition. Okay, so I'm going to walk you through. These are the data that she got. Now, first of all, another former postdoc in our project, Sydney Glassman, she isolated 10 dominant fungal species from a nearby grassland. Charlotte put each of these species in a different microbial cage on sterile litter. Then she put the microbial cages out in areas that were experiencing drought versus ambient water conditions. After a time, about a year, she collected them and then measured how well do they tolerate the drought, how much resource acquisition did they invest in, what was their potential enzyme activity. If there's a trade-off, there should be a negative relationship between these two. Okay, and this is what she found. So I'm going to explain the setup of these graphs first. So on the x-axis here is drought tolerance. And that's what she measured in the field. How well could the fungi grow under drought compared to ambient? This is an effect size. So the larger and more positive a number there is, the more tolerant for drought stress the fungi are. On the y-axis is potential enzyme activity. So how much enzymes are these fungi producing to break down either chitinases, cellulases, or hemicellulases? Okay, now each of these symbols here is a different fungal species, one of those 10 fungal species that we have isolated from the sites. Okay, so you can see here across these three different enzymes, in general, the species that tolerated drought better had less capacity for potential enzyme activity. Okay, so they were doing better at tolerating this drought, but at the cost of being able to break down organic material. Okay, so this is a sign that these evolutionary trade-offs might be happening. But on the other hand, these are just correlations, okay? And we all know that correlation does not necessarily imply causation, all right? So just because a fungus develops drought tolerance doesn't necessarily mean that it's going to reduce potential enzyme activity every time. This could be potentially a spurious relationship. The best way to test whether we do have trade-offs is to actually evolve the fungi under drought and then see, all right, do they lose the ability to acquire resources? Okay, so how do we do this? How do we experimentally evolve fungi in the field? Well, we can take advantage of this microbial cage technique and modify it just slightly. So we can make what we call race cages out of this nylon mesh material. The cages are very long and are narrow. And we can fill them with sterilized litter. And then what we can do is add inocula from a particular fungal species to one end of that tube on the litter, and then set it out in the field. The fungus will grow across the tube, making spores and hyphae as it goes, and then reach the end. By the time it reaches the end, it may have gone through many thousands of generations. So it could have evolved a bit in response to the new drought conditions. Then we collect those race cages, take out some of the inoculum from the evolved end of the cage, transfer it to a new race cage, and start the process again in that cage. All right, then we take another race cage, pull the inoculum out the end, put it in a new one, and just keep it going. All right. Now we just keep this up, keep transferring the more adapted fungus to a new race cage for as long as we can, basically. And we're expecting to do this for 2 years, but if we need to go for longer, we'll go longer. Our a first year PhD student in my lab, Melanie Hakopian, is doing this, and she has set up the cages and set them out in the field. Similar techniques are used in evolution experiments in the lab, except we'll have like glass race tubes with agar in them instead of these microbial cages. And we've had it work quite well in the lab in the past, but again, we need to know what's going on in the field. It could be quite different. So we turned to a local global change experiment. It's about 20 minutes away from our campus, and it was established by Mike Golden, and he continues to maintain it. This is the Loma Ridge Global Change Experiment. And so it's set up, there's a set of plots in grassland and another set in shrubland, which you can't see here. Now, Mike and his group, they built these canopies, very, very large canopies over half the plots. And right before rains, they'll close the canopy, they'll bring plastic over this frame here, and that will divert the rains from these plots. So it's simulating drought. And he closes these canopies over half of the storms, so they're reducing precipitation by about half. Now, this experiment has been going on quite long, and we have a lot of data from it, so we're excited to continue working on it. Now, here's another view of the Loma Ridge Global Change Experiment, and this is from the air. This was an image that was broadcast, I think, on national news. We had a big fire here this last October, right near Irvine. It was kind of scary, but it stayed mostly in the open areas near us. Now, unfortunately, it did completely burn down our experiment. So our experiment is about right there. Okay. Now, I never really expected to watch one of the experiments that I was involved in actually go up in flames on the news, but I did, and so did the rest of us. We are not happy about it at first, but we tried to adapt. And what we've done now is we have changed this experiment to a fire experiment. So we're tracking how the fungi survived the fire immediately after the fire when it was still smoldering and going forward in time. Okay. So that's great, but it ended up not being a suitable place for us to test drought because everything is burned down there. Luckily, and this was before Melanie put her cages out, we were able to switch to another nearby drought experiment. And this is actually right on the UCI campus and some open space we have. This is UCI DroughtNet. It's a member of an international network of drought experiments. We follow their standard procedure for manipulating drought. In this case, we put up a framework with slats here that cover about half the area of the plots that are under them. And so that diverts about half the water away from the plot. Okay, and now this is relatively recently set up, but it was in good enough shape that we could get Melanie's cages out there. So we're actually measuring evolution to drought in this experiment, but it is another nearby experiment. In many ways, it's similar to the Loma Ridge community. So we're very lucky that we had a backup. And so here is the microbial cage that Melanie set out into DroughtNet. She picked 3 different species of fungi to evolve. They each are particularly good at one leg of that triangle, either high yield, resource acquisition, or stress tolerance. So we're going to see whether these 2 here develop better stress tolerance at the expense of high yield and resource acquisition. And I'm also interested to see how this species evolves. It already tolerates stress well. Maybe its tolerance for stress will go off the charts as it starts to evolve. Okay, so Melanie just had her first pickup. She went and checked after about a month. Some of the fungi had already reached the ends of the tubes, which is great. We had a bunch of rain, so that really helped. So we're gonna see how it goes. Now, just for prediction, this is what we expect will happen. If we have here on the x-axis the number of generations exposed to treatment, and this is the start of putting the cages out in the field, we expect that over time, each of these three microbial species should develop better stress tolerance if they were incubated in the drought plots, whereas there might not be much of a change if they're incubated in the ambient plots. Now, looking at other traits that they may have, those species, if they've developed better stress tolerance and there's a trade-off, we might expect them to have less resource acquisition or biomass yield under drought because there is this trade-off. And on the other hand, if they're in ambient conditions, there might not be much change in these traits. Okay, so after these fungi have evolved and we have confirmed that they're tolerant to stresses is different, we're going to collect them. We're going to do the whole gamut of genetic analyses. We're going to do whole genomes. We're going to look at the transcriptomes. We're going to measure their physiology. We're going to get all the information we can. We're very interested in what sort of traits might be tied to evolution of stress tolerance. And overall, I'm very excited to see what we find. We're going to be able to hopefully come back and answer the question, will adaptation to drought lead to reduced decomposition and CO2 production? And if so, how quickly? Right? We're really lucky. Melanie is at the very beginning of her PhD. So if it takes 4 or 5 years for them to evolve, we can do it. We're up for it. Maybe they'll evolve more quickly, but that timeline is going to be very important to know for our models. And then we'll end up putting all this information into Steve Allison's trait-based model called DEMENT. All right, and that ends my talk. I hope that you enjoyed it, and I really appreciate everybody who's collaborated on this talk, and DOE for funding the research, and you for listening, and also the fungi for being great to work with. Okay, thank you.